And this leads us to the last okay. And this leads us to the last chapter of our lecture today, which is the motor proteins. Let's see if it makes it to the next slide. Yeah. So, okay, motor proteins. So the main motor proteins we will talk about, well, the only motor proteins we will talk about today are kinesin and dynein. So, well, that's not true, is it? We're going to talk about myosin, which transports cargoes along acting microfilaments, while kinesin and dynein work on microtubules, but in opposite directions. So the myosin I'm talking about is exactly the same one that you find, well, it's not exactly the same one, but it's similar to the one that you find in the muscles. It works in a very similar way, but it has a slightly different structure because in the muscles, it has the job to generate linear labor, while here it's involved into literally walking onto the actin filament. And also kinesin and dynein, to be honest, they all have a very similar structure, which we will talk about in a second, and they will use the hydrolysis of ATP to move and walk on the filaments. They're proper nanomachines. Okay, a nanomachine is a macromolecular structure which converts chemical energy into movement and labor at a nanometric scale. So kinesin and dynein move things from and to the nucleus, from and to the periphery of the cell, with dynein, which moves cargoes, especially endosomes and lysosomes, from the periphery to the nucleus, while kinesin moves cargoes, usually secretory vesicles, from the nucleus to the periphery of the cell. And this is due to the fact that the polarity of actin filaments is always the same, with the negative sign towards the nucleus and the positive sign towards the periphery of the cell. So a clear example of how kinesin and dynein work is the tadpole. So in order to survive, tadpoles found a way to distribute their pigments across their body. So without expressing all the time the pigments, they will move pigments away from the nuclei of cells during the night. So the pigment will make the tadpole look black and so better camouflaged at night, while during the day the pigment will be transported from the periphery of cells to the nuclei of cells, and so the tadpole will be more translucent and more camouflaged during the day. And this is due to the movements of kinesin and dynein, respectively. So they're also involved into the movement of neurotransmitters. So, I mean, you have to consider that neurons can be very long, but, tiny structures. Think about the motor neuron. It has a cell body which is located in the frontal cortex. It has an axon that runs all the way down to the spinal cord. So we're talking about an axon that has the diameter of less than a micron, or around a micron, something like that. Yeah, let's say 1 micron, okay? Because if the cell body is around 10 micron, we can say that the axon is around 1 micron thick, maybe less than that, but it's long, half a meter. So if you wait for a protein that is synthesized, well, it's not synthesized in the nucleus, but it's synthesized, for example, on the endoplasmic reticulum close to the nucleus, and then for the vesicle containing that protein to move by simple diffusion along the axon, you will wait for a long time. And we need these proteins to be ready, pronto, on the presynaptic side of the synapse. And so that's why we do have these railways of actin and tubulin along the axon, which allow anterograde and retrograde transport. And as a matter of fact, herpes uses retrograde transport. It hijacks the retrograde transport of trigeminal ganglia to move from the epithelia of the mouth or of the nose to the trigeminal ganglion for herpes type 1, while the herpes type 2 can move from the um, anal genitalia epithelium to the sacral ganglion using the retrograde signal, the retrograde transport system, of dynein dynectin complexes and myosin complexes to move from the periphery to the soma of the cell. So these systems are important both in physiology and pathology. And so here you can see also an example of how kinesin and dynein are actually used to move chromosomes in a cell. So motor proteins work by using the usual structure to function relationship. They're characterized by two heads, which are able to attach the tubule or the filament. So in this case, we're looking at the structure of kinesin, which can move on microtubules. And the two heads can attach to the microtubule, then a stalk, and finally a tail, which can attach to the cargo, in this case, the transport vesicle. And the two heads will be able to attach or not and to move or not depending on what they're binding. So for example, if kinesin is binding ATP and it hydrolyzes ATP into ADP and phosphate, then the second head will be able to attach to the microtubule. And then when a new molecule of ATP will bind onto the second head, it will make it less sticky, and so it will detach for the microtubule and it will move forward. And here you see basically another representation of what I just said, right? We do have ATP in green, and then one phosphate will be hydrolyzed, so it will detach, move forward, more ATP will attach, and then ADP will be released, and the head will attach again. So here is an example of how motor proteins can move, but I will not show it in the video because it will stall it and freeze it and destroy and make my computer explode. So I will just upload it with the presentation on Blackboard, and you can see it separately. So myosin works in a pretty much similar way, and you already do know how the two heads of myosin can be used for generating labor. Myosin 2 is the one that makes the actin filaments sliding onto each other, generating strength and force. But myosin 5B and 5A can be used to transport a cargo because of their different conformation and structure, but they're pretty much based on the same principle. And so in 2013, people who discovered the motor proteins, Rothman, Sheckman, and Sudoff, won the Nobel Prize because of it. So to sum up what we've been talking about, cells contain many different kinds of membrane, which are asymmetrical and contain different lipids and proteins. They are also electrically charged, and this is a concept we will talk about again. And they're capable of a variety of transport molecules in and out of cells and their relative organelles. Proteins can be moved around the cell by a variety of transport mechanisms, such as gated vesicles and transmembrane, while the signal sequences determine where a specific protein will go in these compartments. And finally, motor proteins can move cargo along the microtubules, such as kinesin and dynein, or microfilaments, such as myosin. Thank you very much, and see you next time. Good luck and stay safe. Bye bye.